Hello and welcome everybody to another exciting episode of Sci Talks 2.0. As always, your host, Dr. Seksenyan, and today's topic, brain tumors. Today I want to spend some time talking about brain tumors and some of the research advances and discoveries that have been made. I will focus particularly on one discovery that was made recently that allows neurosurgeons and doctors to overcome one of the biggest hurdles facing the treatment of brain tumors. In order to understand some of the hurdles and the research that's being done, I want to give a quick background as to what causes brain tumors and how they're formed. Brain tumors, just like any other tumor in the body, is caused by a mutation in its genomic content. What's the genome, you ask? Well, the genome is the information that a cell needs in order to survive. So if we look within a neuron, what we'll find is that it contains a molecule called deoxyribonucleic acid, or DNA. This DNA contains the information that the cell needs to produce all of the machinery that is required for it to sustain its life. And when a mutation occurs within this DNA, it causes the cell to start growing rapidly and uncontrollably. Among the various causes of mutations is a molecule called a free radical. Free radicals are formed through the natural metabolism that our body does when we break down food for energy. And when these free radicals interact with the DNA, they cause nucleotide changes. The nucleotides are the smallest unit of these DNA, and as a result, when these nucleotides are altered, it changes the whole code of the DNA, and as a result, the cell loses some of its function and therefore becomes uncontrollably aggressive and starts to grow. And as the cell acquires more and more mutations by other molecules that can cause such mutations, it starts to lose complete control and begins to grow and starts to overtake its environment. As the cell continues to grow uncontrollably, it acquires more and more mutations. Consequently, it loses all of its ability to control its growth, and this in turn results in what we call a tumor. So environmental triggers or the genetic causes that started off the initial harm could have been done years prior, and over the period of time when the cell acquires more and more mutations, it eventually leads to uncontrollable growth. And this leads to many different problems and challenges that need to be overcome when we try to treat brain tumors. One of the biggest challenges is that the cells, because of these mutations, acquire what's called heterogeneity. So if you look within a tumor, you're going to find cells that are very different within each patient. And as a result, each of these cells respond differently to chemotherapeutics. And this is a big challenge for people who are trying to design medications targeting these tumors. And the reason for that is, imagine if you have 20 or 30 different types of cells. If you give a chemotherapeutic or any kind of a drug that targets some of these cells, then the rest of the cells which are not affected by it will continue to grow. And as a result, you end up playing a catch-up game with the tumor, and eventually, unfortunately, sometimes medicine loses out in that battle. So in order to be able to understand the heterogeneity, scientists have started using very exciting techniques of single-cell sequencing. This allows us to be able to sequence each of the cells within a tumor to get a better profile of all the different mutations and the different pathways a cell could potentially take when we give it a drug. And therefore, by knowing these different pathways, in the future we'll be able to attack each of these pathways separately and not allow the cells to continue to grow. That's been one of the biggest advances that is being done in the last 10 years or so, and it's an exciting field of research that is generating a lot of exciting and potential avenues of therapy. Now, the other problem that we face as physicians in treating brain tumors is that the tumor looks very much similar to the normal brain tissue. So when neurosurgeons go inside the brain in order to resect the tumor, they have a really hard time deciphering what's normal tissue versus what's a tumor. Although on MRIs and CT scans we could clearly see these tumors lighting up, when in the OR it's really difficult to tell what's normal tissue and what's a tumor. And the problem with that is you don't want to remove normal tissue because you could end up damaging very important and critical structures in the brain. Therefore, it's imperative to be able to tell the difference between normal and tumor tissue. One of the problems that comes along with that is that if you leave some of the tissue behind that's tumor, the tumor grows back much more rapidly. In fact, there are many studies that show that tumors that have been almost completely resected end up doing a lot better compared to those patients whose tumors were unfortunately not able to be completely resected. And as a result, this creates a very challenging problems for physician. The other problem is that in the OR, when a physician takes out a piece of tumor, what they end up doing is sending that off to a pathologist. Now the pathologist ends up looking under the microscope to see whether it's tumor and moreover what kind of a tumor it is and whether there's enough tissue to be able to make this diagnosis. Now sometimes the pathologist will call back to the surgeon who's waiting in the OR with the patient right in front, telling them, hey, you have not removed enough of the margins, there's still some tumor to go. So the surgeon goes in there and keeps trying to take out more and more tumor. As you guys can imagine, this is a very tedious task because first of all, it's really hard to tell what's tumor and what's normal brain tissue, and second of all, to keep waiting for the pathologist to say, hey, you're almost at a margin where we could feel comfortable we've taken out most of the tumor, takes a lot of time. And we know time is money and money is life. And therefore, this creates a very difficult situation. Do you push yourself as a surgeon to take out more and more tissue, or do you hold back? And that uncertainty creates a lot of problem. Now, again, the additional time that it takes for the tissue to go from the OR to the pathologist and have the pathologist look and tell the surgeon takes about 30 minutes. And this technique has been around for almost 100 years. Now, wouldn't it be exciting if there was a way for a surgeon to be able to know whether the tissue that he or she took out was tumor or not tumor, and be able to say instantly whether they should continue resecting more and more or stop because they've reached a border where there's normal tissue. Now, this is the challenge that the paper I want to really briefly discuss about has been able to overcome. They used two very exciting techniques, combined them together to be able to develop a very fast approach that the surgeon could use while operating to instantly, on an order of milliseconds, be able to tell whether it's tumor or not. So how did these researchers develop this technique? First, they used what's called Raman Spectroscopy. Here, if we look at the tissue, what we see is that this is a brain specimen that was taken out of a patient. It has a certain kind of an architecture because of the different kinds of tumor cells. Each of them have a different shape, and there within it is some normal tissue, and as a result it takes on a certain architecture which is not noticeable by eye. But if we use a staining, which is called hematoxylin and eosin, and this is what pathologists do, you'll see that there's a different, slightly different architecture between normal versus tumor tissue. But through the use of Raman spectroscopy, the surgeon could see this almost instantly without having to send this tissue to the pathologist and have the pathologist do the staining, look under the microscope, and give a result back. So what is Raman spectroscopy, right? Raman spectroscopy is essentially using a laser to hit this specimen that was taken out of the person's brain, and by hitting it with the laser is able to essentially highlight out these architectural differences between tumor tissue and normal tissue, and based on that one can say it's tumor versus normal, right? And Raman spectroscopy is a very, very powerful technique, and these kinds of lasers have been approved by the FDA for various other reasons. And so these researchers took advantage of this to be able to develop this technique. Now, the extra added exciting stuff that they did in order to expedite this process of being able to diagnose intraoperatively is they used what's called artificial intelligence, or more specifically neural networks, to be able to make the diagnosis almost instantly. And how did they do this? Well, what they did was they took over 2.5 million different specimens from various different patients, and they trained a computer to be able to recognize some of these subtle differences between normal tissue and tumor tissue. And by being able to recognize these subtle differences, the computers were able to really precisely, almost at a 95 to 99% confidence, be able to tell what's tumor and what's not tumor. So combining these two together, how does the process work? So when we shine light on top of the tumor, because the tumor cells are different in its architecture, we see these bubbles, you can imagine, of wavelengths of light being reflected back. And by studying the different wavelengths of light that is being reflected or being absorbed by these cells, the Raman spectroscopy is able to tell us what's tumor or not. So instead of having a person sit there and say, okay, this is what the Raman spectroscopy is telling me, these investigators trained a computer to do that. So if we put this all together, what ends up happening is the surgeon takes out a tumor, quickly puts it into this machine, shines a laser, and the laser result is quickly read by a computer, and the computer says it's tumor or not tumor. So for example, if we were to look at this brain tissue, you could see that it's really difficult, and unless you're a really trained eye, such as neuropathologist, you can't really tell what's normal versus what's tumor, right? Now imagine, without even disdaining, being able to say what's normal or not under a microscope. But if we take Raman spectroscopy, shine a laser on it, here on the bottom, what we could see is that there's a clear differences in color, and you could really easily see that those highlighted in pink are the tumor areas, and those that are not are the normal tissue. So without even a trained eye, you can tell what's normal and what's not normal. But imagine having the ability to do this within milliseconds, right? So as the surgeon is dissecting through the brain tumor, it knows I'm gonna get almost continuous feedback whether I've gone enough or whether I need to continue dissecting more and more. So this research breakthrough is a tremendous help in order to be able to take out as much of the tumor as possible. In fact, this technology can also be applied to other kinds of tumors, such as dermatological tumors or those within the body outside of the brain. So by being able to prove that this methodology is fast enough, efficient enough, and is specific and sensitive enough to be able to do this, they have opened up the doors to be able to help not just patients with brain tumors, but also those suffering from other kinds of malignancies or cancers. So this research breakthrough has been has created a very exciting fervor amongst physicians treating patients with brain tumors. So this is one of the main purposes of my talk today, to really describe this present paper. and in addition go over just a little bit of some of the other exciting discoveries that have been made and how those are able to address some of the different kinds of dilemmas. Now, I mentioned that the tumor creates a heterogeneous population, and as a consequence, targeting these different cells can become a challenge. So one of the methods that scientists are pursuing is what's called immunotherapy. And the idea behind immunotherapies is through various methods the researchers are using to boost the person's own immune system to start attacking the brain. Now we know that the immune system is involved in various functions to keeping a person healthy, but one of its main goals is to keep you from having cancer. And most people agree that when your immune system begins to deteriorate, that's when cancers start to come up. So when a cancer is present, one of the strategies is to boost that immune system and have it go and do its job. And what are the different methods that scientists are using to boost the immune system? One of them is called CAR therapy, which I will discuss in more detail in a later show. But CAR therapy, just really briefly, involves taking the person's own immune cells, modifying it genetically so that it knows what cells to attack and kill. And once inside the body, the cells can track down these cancers and destroy it. The other one is called dendritic cell vaccination. Dendritic cells are a different kind of immune cells. And what happens is that the scientists take out pieces of the tumor. They then train these dendritic cells to recognize the tumor cells. They then inject these dendritic cells back into the patient, and these dendritic cells then go and inform the rest of the immune system, hey, there's something dangerous happening, and therefore we should go and take care of it. As a third novel approach for treating brain tumors is the use of nanotechnology. Nanotechnology involves coating these small molecules with chemotherapy or any kind of targeted therapy and be able to target them directly to brain tumors. The hope is that these carrier molecules containing all kinds of chemotherapeutic agents can specifically go and target these brain cells and be able to kill the tumor and prevent it from growing or even recurring. So there's been a lot of exciting research and advances being done in the field of brain tumor therapeutics, and the hope is that one of these different approaches will eventually pan out to a drug that could truly wipe out the disease. In the primary paper I just discussed, I have provided with the reference, and if any of you have further questions, please feel free to drop a comment below. With that, I want to thank you for your time and remind you once again to subscribe to my YouTube channel or on Facebook, and don't forget to turn on your notifications for these types of videos in the future. With that, thank you for your time and want to wish you a wonderful day.